All right. Hello, everyone. Well, currently there is no one here in the meeting except me, and that is my fault probably for giving such late notice. But if you're watching the recording on Blackboard, hello, welcome. This is going to be my review session for what we learned this last week and from week one as well. I encourage you at every point that I ask a question to pause the video, think about it, and try your best. Give your answer. This should be interactive in quotations. And hopefully there'll be some people here in the future. But again, that's my fault. Let's see. I have a few questions for you and some stuff that I wanted to talk about. So, genetics, what are we doing here? What are we learning about? This is the point where I invite you to give me some responses. So what are we talking about when we're talking about genetics? Someone might say, am I not sharing my screen? I'm not. Very cool. Let's fix that. That's better. Okay. No, wait, no, I had it. Okay. So genetics, what the heck are we talking about? When you think of genetics, what do you think of? And little hint, we've been working with it a little bit so far this semester. Just to start off with, when I think of genetics, I think of chromosomes, DNA. And so, hold on one second. Okay, sorry about that. Where was I? Yes, genetic chromosomes, that's right. So when I think of genetics, I usually first default to chromosomes. So what is a chromosome? A chromosome is where we find, genetic information. We often see it depicted like this. So this is what we call a chromosome. And a chromosome is usually made up of, well, in this stage, when we see it like this funky little X, what it is two chromatids, two individual chromatids. So this is a chromosome, and these are chromatids. Something's happening with my pen. Oh no, stop that. Scoundrel. Chromatids. I should just write the whole word out. Oh my God, this isn't working. These are two individual chromatids. Oh, this pen. Anyway, these chromatids, as we learned in class, are connected at some point by what is called a, exactly, centromere. Oh my God. Centromere. If the pen would ever stop doing this nonsense to me. They're connected in the center by a centromere. And actually, they're also connected by little fibers called cohesins. But we'll learn more about that later. But this isn't like the simplest part of DNA, because when we think of genes, DNA, obviously, it's in the name. DNA, deoxyribonucleic acid. What we're actually looking at when we're looking at this guy here is, oops, let me fix that. What we're looking at is this little X here. But if we zoom in on this little X, we see that it's a lot of like fiber that's really highly condensed, intertwined. And if we continue to zoom in on like a strand of these individual fibers, we see that it's a super coiling, a very densely coiled structure. And if we continue to zoom in, we find out that the coils are comprised of these things, these little balls. These red parts of the balls are called nucleosomes, and they're just little histone proteins. Each individual ball is a histone protein that connects to this important blue strand And what we're actually looking at when we look at this blue strand is our nucleotide base pairs that we find within that very familiar and beautiful double helix. And so that's what we're actually looking at when, please leave me alone. That's what we're actually looking at when we're looking at these little X's that we see in our textbooks and that Dr. Bell draws on the screen. This is what we're looking at when we're talking about DNA. Excuse me. And so this is our chromosome, and these chromosomes are each made up of individual chromatids. But at different points in the life cycles of our cells, the number of chromosomes that we have and the number of chromatids that they're made up of are different. And obviously we learned about this in class. We talked about it. Well, you learned about it possibly in high school biology, AP. If you took Bio 213 here at GMU, you definitely learned about it. And these are the different cell cycles. And we can also elaborate on this. So usually, we have a cell. It's doing stuff. If you have, say, a liver cell, this whole life cycle, or most of the life cycle, is going to be liver cell stuff. It's going to be full of a lot of lysosomes, and those lysosomes are going to be destroying things that come into your body, your digestive system, and that's what it's going to be doing most of the time. But there is an expanse within the normal life cycle of a cell. That is not good contrast. There is a section within a cell that another cycle is happening. And this is what we call interphase. Well, not all of it is interphase. There are different points along this special life cycle, this special part of a cell's life cycle. These are what we'd call, well, the first, I guess, is what we'd call the G1 phase. G, you can think of as meaning growth. This is where our cell gets physically bigger. If it's a liver cell, it starts out as a little baby, and then it gets larger so that it can fit into the larger liver tissue, the organ, fit into the tissue that then connects to other tissue, which becomes the liver organ. And this G1 phase, like we said, constitutes growth, and it sets up a very important stage that we call the S phase. And S stands for, why is the pen doing this? S stands for synthesis. I have to write in cursive because it doesn't like me tapping the screen. S stands for synthesis. And this gets back to what we were talking about up here, how we have different amounts of chromosomes at different points in the cell cycle. And these are made up of different amounts of chromatids. So the synthesis phase is where we have an individual chromatid. So for a lot of our life cycle, a lot of a cell's life cycle, its chromosomes, will be made up of just a single chromatid. Oh, why does the pen do this? But when we go into this special section of a cell's life cycle, we synthesize more DNA. We create a sister, an identical copy of a chromatid, so that there's more information to be passed along in this later section of this special little blue life cycle of our cell. And so that's what the S phase is. It's synthesis. We synthesize a duplicate of this original chromatid. And these two chromatids together are called sister. They are sister chromatids. They're sisters to each other. Maybe I haven't been pressing down on the screen hard enough. Anyway, they're sisters because this one and this one are perfectly identical. They have the same information, as if they excuse me, came from the same parent. They're sisters because they are identical. And this is important in the next part. So we have our G1 phase that sets up the S phase. And then after the S phase, we have our G2 phase. G2 is another growth phase, and I think of it as setting up the phase that we're all really here for. That is the mitosis phase. And this mitosis phase is where we'll get started with our later questions. But firstly, we can talk about it. Should we talk about it? I think I can write that. Yeah, I'll wait to talk about it. So this is a very important part of genetics. This is what our cells are doing throughout our whole lives. And obviously, that is a pretty important thing. Hold on, I need to check on something. Oh, I have a guest. Hello.
Hello.
I'm glad you're here.
Thank you.
Okay, this is what I wanted to get to. So in addition to these cell cycles that we're talking about, excuse me, or not in addition to, but rather when we're looking at these nice little X's, we don't really care about the X's for their own sake. There's something on the X's that are important. And what do we think that is? Well, we are talking about genetics. What we're talking about are the genes, the special combinations of chemicals that provide instructions for our cells, throughout their cell cycle. Oops, didn't mean to do that. And there's some terminology that we need to know when we're talking about cells. So what we have on this chromosome, one arm of the chromosome, is a collection of genes. Now, what is a gene? It's kind of difficult to understand. We can't really see genes, but genes are, we're all familiar with the set of nucleotide instructions that our cells are able to interpret throughout some special processes that we'll learn about later in the semester. They interpret these single chemical instructions as, instructions, and they do stuff with them. But these genes are not the only kinds of information that we can find on a chromosome. If we have this red gene can also be described perhaps as a pink gene, a lighter red gene. And these differences, the ability for a chromosome to have different variations of a single gene, is called an allele. These are two different alleles. And thinking about chromosomes and genes and alleles, you can think of these kind of like a sentence. In a sentence, you are speaking, or you have words that each represent an idea, and together all of these words can create a sentence that convey a specific meaning. And similarly, we have chromosomes that are made up of genes that convey meaning to our cells. So a chromosome is like a sentence, and the genes are like the words that we use to make up those sentences. And there can be different versions of a word that convey the same meaning, just like there can be different alleles that represent the similar gene. So if I want to say, I love cats, this sentence is like the chromosome. And each individual word is like a gene. It's a specific set of letters or instructions, just like genes are made up of letters like A's, T's, C's, and G's. If this pen would stop messing with me. And the word love is like an allele. You can probably think of synonyms to the word love that convey a similar meaning. So you could say, I adore cats. This sentence still conveys the same meaning, that I'm very fond of cats. However, I could use a word and a synonym of that word, just like a chromosome can use one allele or another allele and still code for the same gene. And that is a very good start to what we're doing in genetics. Sorry. Dun, dun, dun. I will uncode it. Sorry, I didn't prepare this very well. So yeah, this is something I wrote up. It's the essence of what I just said. I will also share this document, so don't worry about that.
Yeah, that will be great.
Oh yes, that's something I failed to mention. Going back to all of this stuff, when we look at a chromosome, the gene isn't just like anywhere on the chromosome. We often find particular genes in a particular place on the chromosome, a specific location. And this location we describe the location using the Latin word loci, or excuse me, locus, whose plural is loci. So this is a single locus. These two together are loci, because they're different locations on this chromosome. Oh yeah, and when we were getting into chapter 3, which we won't talk about in this review, we started getting into Mendel and talking about these things called phenotypes and genotypes. Those are kind of long alien words that could seem very unfamiliar to us if we aren't familiar with genetics already. So what is a genotype and a phenotype? A genotype, you probably recognize the similarity. Genotype, it has to do something with genes. A genotype is when we have a chromosome and all of these alleles, or all of the genes that are represented by specific alleles on that chromosome in specific locations, loci. All of these things together constitute an organism's genotype. The genotype is the concert of all of our, genes represented by different alleles at different loci. And these genotypes, the genes that we're made up of, obviously we can't see them, but somehow they still construct our bodies, and they allow us to do these amazing things as animals. And obviously we can infer that one comes from the other based on intuition. But the more formal way to say this is that we have a phenotype. A phenotype is the visible manifestation, or even the chemical manifestations of the instructions given to our cells by our genotype. So our genotype informs our phenotype, and our phenotype is what we can see and do. Once again, I will share this whole document, and you can maybe even ignore this video and try to figure it out on your own, and then come back to the video and confirm your answers. Also, future reviews will be less sporadic, and I will be much better prepared. I won't be fumbling over my sentences. So now that we have that nice little base to work with, there is actually more base that we need to scaffold to understand what we're really doing here in genetics. So to start with that, let's answer a question. You are a germ cell. Despite the difference in how you're made, what you do, and what you're made of, people often confuse you for other kinds of cells. What distinguishes you as a germ cell from somatic cells, sex cells, and stem cells? So firstly, before we compare ourselves to other things, let's figure out what a germ cell is. A germ cell is, actually, no, let's not do that. Let's just go over them. It's very nice and plain. So a somatic cell, you will see often this zome, like in chromosome, you'll see that prefix, and it means having to do with a body. So somatic cells are our body cells, like our skin, our bone cells, the cells that aren't for reproduction, which is what our germ cells are. You would find germ cells in your sex organs, your testicles, or your ovaries. And these produce our sex cells. So I kind of gave it away. Sex cells, things like our gametes, sperm, eggs, gametes, sex cells, these are all synonyms, are made in germ cells. Or excuse me, germ cells undergo a specific process that we have talked about extensively and we'll probably continue to talk about in this review. Germ cells undergo this process, which we know as meiosis, to create sex cells or gametes. So somatic cells are our body cells. There are our heart cells, our stomach, the walls of our stomach cells, stuff like that. The things you find all throughout your body. Our germ cells you find, they're directly responsible for reproduction on a macroscopic scale, because obviously our somatic cells reproduce via mitosis to replenish themselves or replenish their fallen comrades, but germ cells are for macroscopic organismal reproduction. So what about stem cells? Stem cells are just, well, they're special, and they're kind of a mystery. Stem cells, actually, somatic cells are somatic because the genes that were given to the cell in order to grow it, to build it, determined it to be that kind of cell. Our body needed some more bone cells. Our body needed some more glandular cells. And so they were determined by some mechanism to become a somatic cell. Similarly, our germ cells were told to become germ cells. Stem cells are cells that have not yet been told what to do. They are what we call undifferentiated. They haven't been told the difference between a somatic or germ cell. And therefore, they haven't undergone the process of differentiation to become either a somatic or germ cell. So knowing that, we can move on to the next question. So which of the following cells would you expect to be capable of mitosis? Take a second, decide for yourself. So we just talked about it. Germ cells use meiosis to create sex cells, and somatic cells use mitosis to replenish themselves. So the trick to this question is just being able to identify what is a somatic cell and what is not. So a cat's skin cell, I would say that is a somatic cell. Skin, it belongs to the gross body, and therefore it undergoes mitosis to replenish itself. And skin cells undergo mitosis a lot. They spend a lot of time in that. I wish I hadn't gotten rid of it now. Why can't I do the thing? Let me do the thing. It's not going to let me do the thing. Oh, God. Oh God, I have to fix it. Um, okay, that's better. A little bit. Hey, please stop. Ugh. Okay, we all good, Mike. Let's start sharing. So, oh, damn it. Okay, here we are. Remember, we were talking about this special, it was different colors earlier. Um, this special mitotic stage within the larger cell's life cycle. Skin cells do that a lot because, you scratch yourself. You need to stretch your skin often. Skin cells replenish themselves often because they take such a beating in order to preserve uh, the barrier between our internal environment and the external environment. Anyway, moving on. Sorry for speaking so extendedly. What about a flowering plant's germ cell? Well, we did talk about it. Oh, sorry, didn't mean to click that. Germ cells do not utilize mitosis. Germ cells utilize meiosis in order to create distinct sex cells. These gametes will go on to combine with another sex cell from preferably another organism, and they will grow into a zygote that is actually diploid. Oh, we haven't talked about ploidy yet. Oh, I should do that. Anyway, a cell in a mushroom's mycelium. What do we think about that? I think that this was kind of a trick question. Oh God, I have to find it again. Dang it. As I was saying, leave me alone. Scroll down, please. Oh, I even had this prepared. I'm sorry. It's messing with me. Dun, dun, dun. So how's your day going? We're almost there, I think. We're almost there. There we go. So mushrooms are kind of funky. They have what is called an extended dikaryotic phase of their life cycle, where they aren't the usual diploid that utilizes mitosis. They have some funky nonsense going on, but the long and short is that I don't think that they would utilize mitosis because they're doing some business of having two uncombined haploid cells within a single cell. It's funny. You should take biodiversity if you haven't. You'll learn about it there. Okay, moving on. A human's neuron. Is a human's neuron a somatic cell or a germ cell? I would say that it is a somatic cell. It helps to comprise our body. Now, a bee's exoskeleton. What do we think? This is also a trick question. I'm sorry for tricking you twice in a single question. An exoskeleton is actually the secretion of epithelial cells. So you'll find some sclerotized or generally cuticular secretions that come from epidermal cells in insects like bees, arthropods in general. And because it's not actually a cell, it would not be capable of mitosis. Sorry for tricking you. Moving on. The blood of many animals is red due to the presence of a protein in red blood cells that has an affinity for oxygen. In humans, the production of alpha hemoglobin being that protein, is demanded by the genes HbA1 and HbA2, which are found on chromosome number 16. And this wording that I use, that the production was demanded, it could be confusing, like why did he say that? I like to say demanded just to get across the idea foundationally that our cells don't really want to do anything. They are compelled to do things based on the instructions that they find or are given in the nucleus. in the genome contained within. So anyway, are HbA1 and HbA2, the genes that we talked about, are they homologous or are they alleles? This, I'm so sorry for tricking you guys so often. This was another trick, as testing your knowledge. So homology, we haven't talked about it yet, and I will, I promise. We've heard of homologous chromosomes, though. I was just going to elaborate on it later. So we have homologous chromosomes, and it is on chromosomes where we find genes such as HbA1 and HbA2. So genes are not themselves chromosomes and therefore not able to be homologous. And nor are they alleles. HbA1 is a specific gene that we'd find here. I'll just call it 1. Oh God. And then HbA2 is like another allele, another version. Oh wait, actually no, I messed that up. Dang it. And this is HbA2 down here. Sorry about that. They're different genes. We find them at different loci, different locations on a chromosome. And they don't contain, the same information. They create two different, sorry. I'm confusing myself. So we have these two genes. Genes are represented by alleles, but they aren't themselves alleles. We could have on the HbA2 gene a yellow allele or a dark red allele. So I definitely fumbled that one a little bit, but I was trying to trick you. We just need to understand the difference between chromosomes, which is where we find genes which are represented by alleles. And just so that I can articulate it, because we have had quite a few problems in class talking about the different cell cycles, which we're about to discuss further, but before we do, I just need to articulate a phenomenon, or two actually terms that we have been struggling with as a class a little bit, and that may not have been articulated the most coherently. And that is the concept of a homologous chromosome. Homologous chromosome. So this is like a really long phrase, full of long words, but we can break it down so that it's not so intimidating. So we have two chromosomes, this pink one, or lavender, I guess, and this pink one. These two are homologous. So let's harken back to what we learned at the beginning of the review. What is this individual unit called? That's right, it is a chromatid. Why does it do this? A chromatid. And what is this one's relation to this structure? These two, as we also articulated earlier, are sisters. They're sisters because back in the S phase, where we synthesize new DNA, this one over here duplicated via DNA replication, which is a process we'll explore later in the semester. It duplicated itself and created a second chromatid that is perfectly identical to it. And we call these kinds of chromosomes sister. They're sister to each other. Now, we have these two chromosomes. We have a chromosome that is made up of two sister chromatids, and we have another chromosome that is made up of two sister chromatids. So we know the relation of this one to this one, this structure to that one, but what is the relationship of this chromosome to this one? These two are homologous. And being a homologous chromosome, we can break down the word using etymology. Homo means same, and logos, log is what we're paying attention to, is like information. So homologous chromosomes are homologous because they have the same information. They have the same genes on them. And I could draw this a little bit better. So let's say we have these two chromosomes. I'm going to draw them both as green to convey that they're both, the same information. And we know that on chromosomes, we find genes. So let's say we have this red gene, or no, I'm not going to say that. We have genes, and the genes are represented by these red lines. We have genes that are represented by these blue lines, and we have genes that are represented by these kind of orangey looking lines. You might notice that they're not exactly the same color. So we have chromosomes and there are genes on them. And at different locations on these chromosomes, we have similar genes. So this location, which we call a locus, we'll call it locus 1. This one is locus 2, location 2, and location or locus 3. So the reason that this light green chromosome is homologous to this dark green chromosome is that we have the same genes, the same gene at the same location, same info, and the same holds true for location 2, locus 2. But what we see at locus 3, let's pretend, or let's imagine that orange and yellow, they represent the same gene. Let's say it's the length of your femur, the big bone in your thigh. These both represent the gene that codes for the length of your femur. We notice that this gene, they aren't exactly the same. They aren't both orange or they aren't both yellow. They're different variations of the gene that codes for femur length. So what would we call these? That's right. These are two alleles of the same gene, the gene that codes for, femur length. And this is what it means to be homologous, to have the same information in the same locations on your chromosomes. You don't have to be perfectly identical to your homologous chromosome, just like this light green chromosome has the orange variant of femur length gene, and this dark one has the yellow variant of femur length gene. But you have the same genes, femur length is located at this locus on the chromosome. So they're homologous to each other. And actually, this is a great segue for another thing that we really had trouble with in class, and that is what it means to be diploid versus haploid. And I actually spent a lot of time being wrong about this myself, so no worries if you weren't perfectly clear on it before now, but hopefully we can get it together, I guess. And I'm a fan of breaking things down etymologically. So we know that you'll find me doing that a lot throughout the semester to make words more palatable, understandable. So we know that di is the Greek prefix that means two. Ploid is also a Greek word, and it means folds, technically. So two folds, and folds refers entirely to chromosomes because, it's kind of like folded up and windy and wonky. So two folds means that we have two chromosomes that represent or that have the same information on them. So being a diploid is entirely reliant on whether or not a cell has a homologous pair or has homologous pairs within it. Because, we have more than one homologous pair within our own cells. We have 23. And haploid, conversely, hap I think of as meaning half. So half ploid, half the number of folds. And this is entirely in relation to the diploid number. So for example, you could plants are commonly not even diploid. They could be triploid, 3N, or tetraploid, 4N. So if the normal body cell of a plant cell is 4N, its haploid is 2N because it's half of the number of genetic information relative to the amount that it usually has. And should I save it for question 6? I will save it for question 6. Okay, getting on with the questions. That was hopefully some good foundation. Let's go to number 5. So I'll give you a second. Please match the genetic material that you find in this first column here, A, B, C, D, E, to the phase in the cell cycle where you'd find it. Feel free to draw them out. Okay, so we are kind of just relearning everything. I'm fine with that. In future review sessions with more people, hopefully we will have our own questions and we won't rely so heavily on my questions. I am much more happy to correct your own understandings and help you fix your own mental models than I am like reiterating all of the content that we've already learned in class, which I don't mind doing. You know, it's helpful. Some people may not have caught everything in class. Some people may not have come to class, and we may not have the same like backgrounds. Maybe we took cellular biology at another university and didn't get the same content. Maybe we had another professor that just wasn't as cool as Dr. Bell, and that's fine. So I am perfectly fine reiterating what we were introduced to in class, but I think it would be more helpful to, instead of bringing up the base, I'm not going to say that. Anyway, getting on with the cell cycle, we talked about, that nice little mitosis that we find. So let's talk about mitosis, that one little section that we find. Actually, I'm not going to use that color because it's going to be a bit difficult to see, that we find here in the special part of interphase. So mitosis, we're familiar with it. We have prophase, metaphase, I said metaphase, telophase, and cytokinesis. I'm also going to make those bigger. This should be very familiar. I shouldn't spend too much time on it, but I would like to mention in prophase is the beginning here. We have had our G1, right? Or no, excuse me, our G2. And we have had our synthesis phase. So would we expect in prophase of mitosis to find chromosomes that are made up of 1 or 2 sister chromatids? Which do we think we'd find? Exactly, because we're in mitosis past the synthesis phase. Our individual chromatids have already duplicated to create their sister, and now chromosomes are made up of 2 sister chromatids. So we would find genetic material within our cells that looks like this. Those little X's, the chromosomes. This is prophase. So this is where actually our all of our DNA that's like in spaghetti formation in our nucleus, it is in prophase where this condenses down into those beautiful little X's, those chromosomes. And it is also where the nucleus, which once held the spaghetti, dissolves. The pen won't let me just draw dotted lines. So imagine a dotted line surrounding those X's. And And then in metaphase, which follows prophase, metaphase, meta means center. This is where our chromosomes align in the center. We know this. This is also when spindles attach. Centrioles. And then I'm going to erase that. Metaphase, meta means center. It's where chromosomes align in the center. Then we have anaphase. Anaphase is where these chromosomes, which are made up of two sister chromatids, are split apart into their individual chromatids. So imagine like whoosh, they're being pulled toward the pole of the cell by these spindle fibers. This is anaphase. And then in telophase, we have our chromosomes. They have been separated. Actually, this is something that I should articulate. So obviously it's confusing. It's what makes questions like this difficult and what we explored in class difficult. Why is this little guy, why is that a chromosome? But this isn't two chromosomes if it's made up of two of these little guys. The reason is actually really intuitive. It doesn't have to be confusing. So we know that this guy is made up of two sister chromatids, and sister chromatids are identical to each other. So when they're together, this one isn't providing any new information to the cell. Sorry, I didn't word that very well. This chromosome does not provide any new information to the cell that this one didn't already provide. And because they are just identical copies, we treat this as a single chromosome because both sisters provide the exact same information. However, in anaphase, when the sisters are split apart, they become chromosomes in their own right once again. And so that is why at this point, we have 3 chromosomes, which are made up of 6 chromatids. And a chromatid, we often in class call a DNA molecule. I wish my pen would just be nice to me. It's okay. It helps me a lot. I should be nicer to it. DNA molecule, chromatid DNA molecule, whichever you want to say. And so we have here at this point 3 chromosomes made up of 6 chromatids. And at this point, anaphase, when they are separated, we now have 6 chromosomes. They become chromosomes all in their own right, because they are going to provide the information to the corresponding cell. So it is at this point that we have 6 individual chromosomes. And then in telophase, something occurs. Our cell starts to split apart because, mitosis is all about the creation of 2 identical daughter cells from a single parent cell. Identical. And, the reason they're identical is because exactly this one and this one are perfectly identical. They're sister chromatids, or they were sister chromatids. And actually, I'll get to that. Where was I? Oh, yes. So in telophase, we have the formation of a special structure that will help us split apart later. And that structure is called a cleavage furrow. And I think of it as like we have this bump forming Actually, that could be interpreted as another kind of cleavage. Anyway, we're all adults. I think of a cleavage furrow as like someone took a big like butcher's knife and they cleaved down into this and created a little bump. And that's why it's a cleavage furrow, not for any other reason. Yeah, telophase is all about the formation of a cleavage furrow, and then we split into our two distinct cells in a process called cytokinesis, which also sounds like a very alien word. But if we break it down, cyto means cell, and kino, like kinetic energy, is all about movement. So cytokinesis is when cells move, they move apart from each other. And they are now two independent, but still identical daughter cells that are made up of how many chromosomes and how many DNA molecules each? Exactly. They're each made up of 3 chromosomes. We can count them 1, 2, 3. And each chromosome is made up of 1 chromatid. So we find 3 chromatids or 3 DNA molecules. So this is mitosis, very nice and plain. Meiosis is only a bit more complicated. So that was mitosis. That mitosis is something that our somatic cells will do exactly, but in our germ cells, they don't undergo mitosis. Well, actually, I'm not sure, do they? Because germ cells need to replenish themselves too. Maybe we have stem cells that create all of our germ cells. I don't know. I should learn that. I invite you to learn it yourself. However, we have meiosis, which occurs in our germ cells. And meiosis, I'm not going to be very long-winded about this. Meiosis is broken down into parts 1 and 2. Part 1, we have our usual prophase, metaphase, anaphase, telophase, and cytokinesis. However, in meiosis 1, our anaphase and our metaphase are different. In meiosis 1, we're very concerned with the homologous chromosomes that we explored earlier. I'm not going to make those so big. We are very concerned with the homologous chromosomes. So let's say we have these reddish orangish colors, they're homologous to each other. Then we have 2 green chromosomes who are homologous to each other. And also, I'm not going to draw it like that so I can convey some deeper meaning. Actually, no, that is metaphase. So I'm going to draw it that way. We have our green chromosome in prophase, metaphase. Sorry, I'm being so scatterbrained. I've had a long day. Prophase, metaphase, anaphase, telophase, and cytokinesis. I'm going to end up drawing these again anyway, so I may as well just do it now. Dun, dun, dun. Telophase and cytokinesis. And then we have our red chromosome, our reddish chromosome, and our other green chromosome. Prophase, they condensed into their nice, beautiful little X's. And then in metaphase, we know meta means center. So we know that our chromosomes are going to be aligning in the center. But in meiosis 1, chromosomes align in their homologous pairs. Green chromosomes match up to green, reddish chromosomes match up to reddish. And something that's really important to meiosis, because in meiosis, just like in mitosis, we are trying to create 2 identical daughter cells from a parent cell. Conversely, in meiosis, we are trying to create genetically distinct daughter cells from a parent cell. Meiosis is all about creating genetic variation because we find it in our germ cells. Our sex cells are created in meiosis. And so not all of our offspring, it wouldn't be advantageous for all of our offspring to be genetically identical, because what if a plague ravages our population and there's no variation, there's no chance for variation that could possibly give one of our offspring the tools to survive because they're all identical to each other, all identical to me. So what we want from meiosis is variation. Unlike mitosis, where we want, all of our skin cells to be the same. We want all of our heart cells to be the same so that they can beat in unison, stuff like that. And on that line of thought, we have a special phenomenon that Gregor Mendel remarked on when he was first studying inheritance in the 19th century, and that is what is called independent assortment. This occurs in metaphase 1 of meiosis, or metaphase of meiosis 1. And that is where you see that we know homologous chromosomes line up in their pairs in the center. However, it doesn't have to be such that red is on the right and orange is on the left. It could be that red is on the left, or red is on the right, and orange is on the left. And whether or not orange is on the left versus the right, whether or not lime green is on the right or the left, will impact not only the resulting cells of cytokinesis from meiosis 1, but also the 4 daughter cells from that result from the 2 cytokinesis events in meiosis 2. So independent assortment is a phenomenon that occurs in mitosis, very important. And in anaphase of meiosis 1, we don't split our homologous chromosome into their sister chromatids. We instead just break apart the homologous chromosomes from each other. So once again, pretend like whoosh, you're being sucked away. They're actually being pulled away. Whoosh. Homologous chromosomes are pulled away from each other in anaphase. And then in telophase, we have these chromosomes. And how many DNA molecules or chromatids make up these chromosomes? Good job. Each chromosome is made up of 2 chromatids or 2 DNA molecules. Very good. And so we end up with a cell, or 2 different cells. Let's pretend this one went up here, this one went over here. We end up with this and this. So we can already see that if this orange were instead on the left in the place of red, then orange would be here, then here, and it would end up here. So we'd have orange and lime green instead of red and lime green. So we see that independent assortment is already creating some very significant genetic variation for our offspring. And that is meiosis 1. Meiosis 1 is all about separating homologous pairs from each other. However, in meiosis 2, I'm going to keep my colors consistent. In meiosis 2, we have the same phases, those very familiar phases. And in this case, let's focus on the red and green cell. It could be helpful to, in order to visualize the genetic variation that we find. There's another piece of genetic variation or another process that affords our offspring extra genetic variation that we haven't talked about. This occurs in meiosis 1. It's called crossing over. We'll explore it more in, I think, chapter 7, which is recombination and linkage. But yeah, for now, let's just focus on that cell with red and lime green. So we have our prophase, our metaphase, our anaphase, our telophase, and our cytokinesis. And this is for meiosis 2. However, unlike in meiosis 1, we don't have homologous pairs anymore. Our red chromosome was separated from its buddy, its homologous pair. They had the same information. It was so sad. They are no longer together. So because we no longer have homologous pairs, the chromosomes can instead just line up single file during metaphase. Remember, meta in the middle, they line in the middle. They don't have a homologous pair to line up next to. So instead, they just line up in the center by themselves, which makes it such that spindles can attach now to the individual sister chromatids. Sorry, those should be connected at the poles. So now the spindles, instead of just attaching to the chromosome as a whole, they now attach to the individual chromatid, and then they pull our chromosomes apart into the sister chromatids that make them up. And we get them now separated with a cleavage furrow forming in telophase of meiosis 2. My voice is starting to get soft because I'm tired. So what have we noticed here? We've noticed in meiosis 2 that the number of, actually, we'll explore that later once we've finished. So we have our anaphase where chromosomes are split up into their chromosomes are split up into their sister chromatids. And then these sister chromatids are further broken apart and they fall into their own cell, their daughter cell, which is, in a way, unique. And this is different from what we'd get in this cell. So if this were in this place, we'd have an orange here. And therefore, we'd have orange and green instead of red and green. So once again, we see the genetic variation that can arise from independent assortment in metaphase 1 of meiosis. And we end up with these daughter cells. That was a lot. And what we noticed throughout, you don't have to pay attention to this. You could just pay attention to the fact that here in prophase of meiosis 2, we have 2 chromosomes, 4 chromatids in this whole cell, because, you know, we have 2 chromosomes and they're each made up of 2 chromatids. We see that through meiosis 2, we start our cell with 2 chromosomes and we end our cell with 2 chromosomes. However, these chromosomes, the number of DNA molecules has changed throughout meiosis 2. In meiosis 1, we started with 4 chromosomes, 8 DNA molecules, and we ended with a different number of chromosomes. We started with 4, ended in 2. Here we started in 2 and ended in 2. So in meiosis 1, we reduce the number of chromosomes that we have in a cell because, you know, we're splitting homologous pairs apart from each other. However, in meiosis two, we're splitting sister chromatids apart from each other. And when we break them apart, and then break them apart further through cytokinesis, we conserve the number of chromosomes that we have because once this is split apart into its component sister chromatids, the two chromatids become chromosomes in their own right. Oh, my voice is so tired. So now that we have that, we should so very easily be able to answer question number 5. Match the genetic material with when in the cell cycle you'd find it. So when do we condense our chromosomes? Condense
Synthesis.
our chromosomes. Very good. In prophase, it's when we no longer have them in spaghetti, like we talked about earlier. That's so long ago. It's 3 o'clock now. So yeah, we condense our chromosomes during, or I guess prior to prophase. When do we separate our sister chromatids from each other?
Anaphase.
We separate our sister chromatids from each other in, that's right, anaphase of mitosis, or exactly. I had a little sneaky, tricky question there. We not only separate our chromosomes into sister chromatids during mitosis, but we also separate them from each other, as we just drew, in anaphase of mitosis, or excuse me, meiosis 2. There's so many tongue twisters. So anaphase 2, as well as anaphase of mitosis. When do we get 2 unique daughter cells? We get 2 unique daughter cells from mitosis.
Cytokinesis.
We know this. We want our, like say a lizard. I actually, unfortunately, did this to a gecko this summer. If you pull on a gecko's tail even a little bit, if they think you're a predator, some species of gecko will cinch their muscles around a specific point on their tail, and this will cause their tail to snap off or fall off. And so eventually, hopefully, the lizard can regrow its tail. And when the lizard regrows its tail, the lizard's like tail muscle and blood cells that were in its tail, those are somatic cells, which undergo mitosis. And when they undergo mitosis, we want those cells to reproduce via mitosis and create identical cells. We want muscle cells to produce muscle cells and replenish them. Well, technically not muscle cells. I forgot to mention the G0 thing, but we mentioned that in class, so I don't really have to reiterate it. The nerve cells that previously innervated the tail of the muscle cell, when those replicate, actually, a lot of neurological cells don't replicate. They're stuck in the G0 phase. They're blood vessels, the blood vessels that previously vascularized their tail cells. Finally, found a good example. The lizard wants those to also use mitosis to produce more blood cells. We don't want them to produce skin cells like inside of the newly grown tail where we need blood to be circulating. So we want to produce, oh my God, I was talking about the wrong thing. I'm tired, I'm sorry. Moving on to the question or the answer that I was actually talking about. Creating identical daughter cells in the case of a lizard's tail would occur in mitosis because we want the same cells producing the same functions. Oh, goodness. And so where would we want unique daughter cells? We need unique cells in order to produce variation so that our offspring could potentially be more equipped to deal with certain environmental stimuli. And that also occurs in cytokinesis. Remember, cyto means cell, kinesis means movement. So the cell movement, the breaking apart of our cells into 2 daughter cells from a parent cell, we want unique cells from our germ cells, which undergo meiosis. And that happens specifically in meiosis 1. And DNA replication is when we have a chromatid that doubles itself, duplicates, and that occurs in the synthesis phase. So we synthesize a sister. You can think of that mnemonic device. We synthesize a sister. Whew, I'm tired. Now I'm gonna grab some water really fast. 1 second. So now that we've finished giving our background information over the last hour, thank you for sticking with me. It's hard to speak for that long, but it's probably hard to listen for that long too. So thanks for hanging out with me. Now that we're done with all of that background, let's talk about some stuff that we talked about in class. We did this a lot, and actually there was quite a bit of confusion concerning the take-home question that we had at the end of last class. Or no, it wasn't the last class, was it? I think it may have been last class. We were doing it in the middle of last class, where we were given the number of, or the amount of genetic information that you found in metaphase of meiosis 2, and then you had to go through the cell cycles, mitosis and meiosis, and determine the number of DNA molecules in the various stages. So let's do some more of that. I am also totally willing to go over that at the end of this if my voice is not totally gone. So an owl's skin cells, I knew I was forgetting something. This whole business of being 2n equals 82, this is something that every table that I visited during class had a little bit of a shaky grasp on. And I think the incorrect idea, or not incorrect, but misconceived idea. So 2n equals 82, it is so tempting to think of this as an equation to give us the answer. 2n equals 82, it's not an equation that we use to find the amount of anything that we have in the cell. It just conveys information, which I guess that's what an equation does, but this is not an equation. We are not solving for n. N does not equal 41. We can mark that off right away. 2n simply refers to being a diploid organism. Remember, we talked about what it meant to be a diploid versus a haploid versus a tetraploid. And that just has to do with the fact that within their cells, these organisms have one homologous pair, 0 homologous pairs, or 4 homologous pairs. That's right, in plants, they have some pretty funky business going on. And they, unlike animals, are able to deal, I wish I had another color that was similar to orange. Oh yeah, here we go. So a single chromosome within a plant cell, some plant cells, some plants are diploid, some are triploid, some are hexaploid. You don't find many pentaploids, but a plant cell is 4n tetraploid if a chromosome has some homologous pairs equaling 4 chromosomes that contain the same information, the same genes in the same locations as we were talking about earlier. So being diploid, this 2n, you're not solving for n. This simply means that we have a diploid organism, and therefore every cell will be made up, or it will contain homologous pairs. I'm obviously not going to draw all 82 homologous pairs that you'd find in the cells of an owl, but and actually I think this is a great horned barn owl. That's 2n equals 82. So 2n means diploid, and 82 means that we have 82 chromosomes. So within an owl's cells, all of the chromosomes have a homologous pair. And out of all of these chromosomes, homologous pairs included, there are 82 of them. Nice and simple, right? It's just we have to get over the temptation of trying to solve this as if it were an equation. So we could, I could draw this in a less numerous fashion. So let's say we were 2n equals 4. We have a cell, we have a chromosome with a homologous chromosome, or a chromosome that is homologous to it. If we wanted to define the relationship between homologous chromosomes, we could say that this dark green chromosome is homologous to this light green, just as this light green chromosome is homologous to this dark green. 2n equals 4. We have the position within the cell of a chromosome is arbitrary in this drawing. So this cell is 2n equals 4. It's 2n, it's diploid because it has homologous pairing. And there are 1, 2, 3, 4 total chromosomes. So knowing that, we can answer this question. An owl's skin cells are 2n equals 82. How many chromosomes and chromatids will be made up, will be present during prophase of mitosis? So 2n equals 82. What do we know? We know that there are going to be 82 total chromosomes. So we can instantly check off this one and this one. So how many chromatids make up these 82 chromosomes that we find in the owl's cells? Which one?
164.
Well, the answer, see where in the question you can find a hint to the answer. Pause the video to find out. And we know because we are in prophase of mitosis, we know where we are in the cell cycle, and therefore we know how much genetic information is used to comprise the chromosomes that will be shuffled around throughout either mitosis or meiosis. And so we know that in prophase, we can redraw it. This is where our chromosomes condense. They used to be that spaghetti nonsense all throughout the nucleus of our cell. However, that was during the regular life cycle. When we then move into interphase and explore that cycle, remember we had G1, S phase, G2, and mitosis. So we know that prophase of mitosis obviously occurs in mitosis. And before that, we had the synthesis phase. And the synthesis phase is where we previously had chromosomes that were just made up of a single chromatid. However, it is during synthesis where chromosomes undergo DNA replication, and a chromatid will replicate, produce its sister, and therefore a single chromosome is now made up of 2 chromatids. Or 2 DNA molecules. One day I will figure out, maybe the pen is out of battery. Anyway, so if each chromosome during this phase is made up of 2 chromatids, we know the number of chromosomes, so we can simply multiply by 2 to get the answer. That was a bit more tricky than what you'll be asked on an exam, I think, because On the exam, we are largely sticking within the very familiar prophase, metaphase, anaphase, telophase, and you'll usually be told, well, I guess, never mind. We're kind of made to assume that for the example in class 2n equaling 10, we had 10 chromosomes in this prophase that were each made up of two chromatids. And I guess that can be assumed for the rest of the questions that you'll find. But if you're ever asked how many chromosomes and chromatids are present in like G1 phase, for example, before the synthesis phase, that's when you have to know for a fact and be able to repeat for an exam that chromosomes are made up of a single chromatid, a single DNA molecule. Okay, moving on. This is more in the spirit of what we were doing in class. If a daughter cell has 56 chromosomes at the end of meiosis 2, how many chromosomes did the parent cell have? I know the answer to this. And I was talking about it with one of you guys. Like, I know this. Don't feel guilty or stinky or dumb for like having to draw everything out. The only reason I can do cell cycle stuff in my head is because I've been doing genetics for many semesters. So I can just visualize it. But I started off drawing every single stage every single time. Don't feel bad for doing so. And in this question, it's very valuable. So we know that we're in meiosis. We can split this up into meiosis 1 and meiosis 2. And we can name our stages. We have prophase, metaphase, anaphase, telophase, and cytokinesis. So a daughter cell has 56 chromosomes at the end of meiosis 2. So we are talking about this stage right here. A single daughter cell, so this guy right here, for example, has 56 chromosomes. Can you tell me how many DNA molecules that it has? Based on what we know, exactly, by the end of meiosis 2, a single chromosome should be made up of a single chromatid. Good job. That's not a part of this question. I just thought I'd ask. So if we have 56 chromosomes here, how many chromosomes did the parent cell have? Because obviously we had our former parent cell, our germ cell, and then certain chemical pathways engendered it to begin the process of meiosis, to start producing these gametes. And this circle here could be, the sperm cell. This one, an egg cell, you get the point. So if at this point we have 56, how many chromosomes did this cell here have? That's something that you have to be able to recognize within the question, is what we're really asking. We're asking, how much genetic information did this cell here have? And we can work backward. So we had 56 here. Let's say that one little chromatid represents 56 chromosomes. There's 56 chromatids. So we had one here, which came from here and here. So within this cell that hasn't yet split into two, there are currently 2 times 56. So 112. The daughter cell is 56 at the end of meiosis 2. Just making sure I read the question properly. Yeah, so we have 112 chromosomes here. Oh wait, no, that's not right. Oh, that's better. This is anaphase, they split apart. This is metaphase, they were together. Prophase, they're doing their thing. And then here we have them doing their thing. as well as their homologous pair, which was doing, or not homologous pair, the homologous chromosome, which was doing its thing. Oops. Being pulled away, very sad. Metaphase, they align in the center in their pairs. So here we are. I don't have the right answer as an option, but let's talk about it. So we had 56 chromosomes in a single daughter cell. But before they split into their individual cells, I'm going to start using another color because this is already looking like a mess. Before they split into the two daughter cells, we had 112 here still, chromosomes. However, right here, a chromosome is made up of two chromatids, these two that we found here. So this, before they split in anaphase of meiosis 2, was still 56. Means 56 here. Right, dude. Which means 56 here, 56 here. However, before they split apart into their own distinct daughter cells, if each, this is a single chromosome, and there are 56 of them. We're just working our way backward. We have 56 here, represented by one chromosome, and 56 here. So that becomes 112. I'm sorry, I could have chosen a number smaller than 56, and then I could actually have drawn them all. But for the sake of people doing it on their own, I thought I'd give a less uniform example. So if we have 112 here, we also have 112 here. We have 112 here, which means that here we also had 112. And we could reduce this question so that it's evident through the picture that if a cell had two chromosomes at the end, had one chromosome at the end of meiosis 2, how many chromosomes did the parent cell have? If we had one here at the end, and we followed the journey of a single chromosome all the way back, we'd see that at the beginning, through the processes that we described, would have two chromosomes. So we have a chromosome ratio from cytokinesis 2 to prophase 1 of 1 to 2. If we translate that, 56, I am convoluting this very intensely. It is, don't worry about this ratio stuff. Just try your best to work backward. And we get the result of 112 chromosomes. Asking chromosomes, not chromatids. If we were asking chromatids, how many would be the answer? Exactly just 112 times 2, 224. Ooh, that was a lot of talking. Yeah, so we went through, I'm going to get rid of that. So we went through like the basics of genetics, some very foundational ideas and definitions that we will need throughout the semester. We talked about the different kinds of cells and what kind of processes they undergo, which cell cycles they usually enact. We talked more about the kinds of cells where we'd find them. I tricked you and I tried to assess your knowledge of what an allele versus a gene versus a locus is. And number 5, we talked about the meiosis and mitosis cell cycles in great detail. Number 6, we talked more about diploidy and what that funky little diploid signifier is, 2n equaling some number. And then we did a little example, number 7. Whew, nice and long. It's been an hour and a half. I think that this is very foundational, very, I don't want to say, it can definitely be hard, but hopefully through this review, we've explored it in enough detail to make it comprehensible and very easy to follow. So actually, should I go through the example that we were given toward the end of last class? I will leave that up to the one person who is gracious enough to have joined me. Do you have an opinion on whether or not we should? Oh my gosh, my volume has been all the way down on my computer the whole time. So if you've been talking and I have been ignoring you, please forgive me. I'm so sorry.
Oh no, it's okay. I was just saying if it was A or B or C, just saying the answers.
Oh, thank you so much. Oh my God. I'm sorry. Do you have an opinion on whether or not I should go through the example that we had in class?
The last class we did. Well, it's okay for me. I already got the answer, so I
Awesome.
don't mind if you go through them. So
Okay, thank you.
even if you don't, it's okay for me. So
Awesome, thank you. And do you have any outstanding questions? I could talk about chapter 3 if you'd like, or anything else you'd like, because if not, I think that we have done quite enough for this review.
it was actually great. Thank you so much. No, that was all. I have some questions, but you already answered them.
Awesome, fantastic. Then I think that's going to do it. Thank you so much for joining me. Actually, could I have your name so that I could give you the oops token? I should have
Oh.
also mentioned that at the beginning of this recording that join us for review sessions and you'll be rewarded with an extra, we talked about this in the first class, but like if you
Right.
got a bad homework grade or if you missed a quiz, we'll drop that if you come to our review sessions. So could I have your name, please?
First name Hevar, Hevar.
Oh, sorry, yes.
Oh, sorry. It was Hevar.
Hevar. Thank you very much.
Mhm.
And I will definitely record that for you. Thank you so much for joining me. I am so sorry once again.
No, it's all right.
And thank you if you watched the recording. It should be up probably in like 30 minutes. But yeah, that's all I had for today. And does just for you, because you were kind enough to join me, does this 2 to 4 time work out for you, or would you prefer 1 to 3?
Well, I don't have anything to do, so both times are okay.
Okay.
This 2 to 4 is fine.
Awesome. I think next week we will try 1 to 3, just in case, because the survey results were inconclusive in that regard. But yeah, thank you so much for joining me and for watching if you did. And I will see you guys in class on Tuesday. If you have any questions, please email me or email Manar or use the Discord.
Thank you.
Thank you. And once again, I hope you guys have a great rest of your day. We'll see you on Tuesday.
You too. Bye.